Hello everyone, this is your instructor Kwamishen, and this is part one of our lecture on the classification of prokaryotes. By way of objectives, we will be refreshing and reviewing our minds on the major taxonomy of life, the differences between the three domains, archaea, bacteria, and eukarya, the basic categories of archaea, representative groups in terms of the major taxonomic groups that we do have, the characteristics of bacteria, the taxonomic groups, and their representatives as well. So one resource that is heavily relied on, although it's not an official reference, but it's a very authoritative reference for classification of organisms, is the Burgess Manual of Systematic Bacteriology, which relies on the rRNA sequences of organisms. The rRNA sequences are highly conserved regions of nucleotides, and so based on the sequences, we can actually tell how closely related or otherwise that organisms are to each other. So based on that information, we can group bacteria and archaea variously. We'll quickly review some information on how broadly we group prokaryotes. So morphologically, how the microorganism is shaped in terms of whether it's a cocci, is it spherical, is it bacillus, is it rod-like, is it coccobacilli, which is somewhat between a coccus and a bacillus, vibrio, spiricate, or is it pleomorphic. Another important information that is used to group prokaryotes are their manner of reproduction. Prokaryotes reproduce asexually and mainly by binary fission. But there are variations of binary fission that also exist. So the classical binary fission, you would have a bacteria or an archaea. The genetic component will then duplicate and divide, and then the cell, other cellular components would also divide, and eventually you end up with two daughter cells. One variation is a snapping division, where you would have only the inner portion of the cell wall. Remember, the cell wall has two layers, so only the inner layer will invaginate. So the inner layer will then grow inwards and divide and separate the genetic contents. And the daughter cells will then continue to grow. But as they continue to expand, tension will be placed on the outer layer, and then so there is a snapping. So the outer layer then snaps off like that. And so that's an example of snapping division. The cells may remain together for a while, and eventually they will separate. Then there is budding as well, where a portion of the cell will grow and elongate, and then the nucleoid, the divided nucleoid, will then move into the new growth area, and it will continue to extend, and eventually it's going to break off or bud off. So the new cell, which is smaller than the older one, will continue to grow until it becomes a matured one as well. So these are all variations of binary fission, and based on this, we can use them to also group prokaryotes. The cell wall composition is also another important characteristic. There are some microorganisms that do not have cell walls. There are others that do have cell walls, but their cell walls doesn't have peptidoglycan or PG. Those that are gram-negative, for instance, would have a lot of lipopolysaccharides in the outer cell membrane, which is absent in gram-positive. So it's a very important information that is heavily relied on to generally group microorganisms. Using staining techniques, especially gram staining, we can group organisms, prokaryotes, into either gram-positive or gram-negative. Gram-positive meaning they will retain the purple stain, the crystal violet stain, and gram-negative meaning they will pick the counter stain, pink staining, saffronin. And then molecular methods are also used to group microorganisms. I already alluded to rRNA sequences that are used, and so other molecular components in terms of the genetic constitution of the microorganisms are also used. And so a review of the central dogma from genes to proteins is important, that genetic information is stored in the DNA. It will then be transcribed into messenger RNA and translated into protein, and that's how the cycle works. And so the survey that we're doing in terms of this classification, we would first look at archaea. For archaea, the common characteristics of archaea is that they lack peptidoglycan, so the cells do not have peptidoglycan. Their cell membrane have lipids that are heavily branched, but they have a start codon. AUG has a start codon which codes for methionine. Okay. Note that in bacteria, the start codon codes for formyl methionine and not methionine. There are 4 phyla. We will basically concentrate on these 3. And so remember there is a broader group, the domain. So you have archaea, you have bacteria, you have eukarya, and next to the domains you have the phylums, and then from the phylums you have the class, you have the order, you have the genus, and the species. Okay, and so there are 4 phyla, that's the plural. We will basically concentrate on the first 3, the Cranoarchaeota, Euryarchaeota, and Cararchaeota. They reproduce by binary fission or budding. Most of them are either cocci, bacilli, or spiral, but they are pleomorphic forms that exist. Now, when we say pleomorphic, it means that these bacteria or these prokaryotes generally do not have cell walls, and that they're able to have different shapes. So they have various sizes and shapes, and so because they don't assume just one shape, we say they are pleomorphic. There are no known pathogenic members of this group, so they are not known to be causing any diseases as of now. So some noted group members that we'll look at are grouped based on the environment that they exist in, or based on what they produce, the products that they produce. So we have extremophiles, and then we have the methanogens. For the extremophiles, they require extreme conditions to survive. And so the extreme condition may be temperature, maybe pH, and or salinity, the salt concentration. Prominent members that we have in this group are the Thermophiles and the Holophiles. For the Thermophiles, they cannot function. The proteins that they synthesize, their DNA, RNA, and the membranes and the cellular composition generally cannot function properly at temperatures that are below 45 degrees. So they need higher temperatures in order to actually stay alive. And then within the Thermophiles, you have the hyperthermophiles. They will require extreme temperatures over 80 degrees. Some are known to live at temperatures of 121 degrees. Most of them are Cranoarchaeota, but you have some of them that are Euryarchaeota. There are 2 representative genera, the Geogemma and Pyrodictium. An example of the Pyrodictium is the Pyrodictium abyssi, ABYSSI. And this particular archaea is known to live at temperatures of 110 degrees, extremely hot. Those that live at temperatures around 120, 121 degrees, they have this specific shape, disc-like shape, that helps them to be able to live at that temperature. And so we've been able to isolate some of them at very hot areas, the hot springs. In addition to that, you have the Holophiles. So you have the Thermophiles, and within them, the hyperthermophiles, you have the Holophiles. The Holophiles are all euryarchaeota. All of them are euryarchaeota. An example is Halobacterium salinarium, that inhabits very saline, extremely saline habitats with salt concentration around 17 to 23% in order for them to even survive in that environment. And many will contain pigments, which may be red or orange, and most likely will serve as a protective mechanism. So the pigment generally in prokaryotes protects them from very high energy molecules that are produced during the process of photosynthesis, or free oxygen radicals that are produced during respiration. Then you have the methanogens. The methanogens are the largest group of archaea. So you will notice that there are certain characteristics that are coming through, which can be noted. What the methanogens do is that they convert carbon dioxide, hydrogen, and some organic acids into methane gas. So environmentally, they are of economic importance because they can help in converting all these waste into methane in the sediments, deep down in the soil, and the oceans as well. And so we find that they live in the colons of animals and help them, like cattle, help in converting carbon dioxide, hydrogen, and organic molecules into methane. And then they are also known to produce trillions of tons of methane under the sea floor. They are implicated in the greenhouse effect, because methane is a greenhouse gas. So when methane is produced and beyond a certain concentration, they're able to trap the sun's rays once it comes in, just like carbon dioxide, and prevent it from leaving the atmosphere. So it contributes to the heating up of the climate. And then there are uses of it in terms of industrial application, in sewage treatment plants, and the production of methane for use as a gas. So bacteria, however, are quite different from archaea in that they generally would have a cell wall that is made up of peptidoglycan. So we'll group bacteria based on whether they are gram-positive or gram-negative. On the slide that you're currently looking at, all these bacteria, all these are gram-positive. Okay, so these are all gram-positive bacteria. The rest are all gram-negative. However, I'll treat them this way. We'll begin with deeply branching bacteria, and mainly because they inhabit very similar conditions just as the archaea. So I want to treat them right after archaea. Then we'll look at the gram-positives, those that have low content of guanine and cytosine, those that do not have cell walls, and those that have the high content of G plus C. And then we'll look at the gram-negative bacteria as well. So for the deeply branching phototropic bacteria, these are gram-negative bacteria that are not closely related to gram-negative proteobacteria, which are the typical gram-negative bacteria. The deeply branching are similar to the earliest bacteria or the archaea. They are autotrophic, So they produce their own food using energy, either from the sun's light or inorganic substances in the environment. They live in extreme environments. They may be very hot, they may be very acidic, or there's a lack of oxygen or very high radiation. Representative groups are the Aquifex pyrophyllux, and that's the picture of the Aquifex pyrophyllux. You see the flagella in there. They represent some of the earliest branches of bacteria to have existed on Earth. Then you have the Deinococcus radiodurans, which is a very interesting bacteria. So the outer membrane is similar to gram-negatives, but they stain positive. They are extremophilic bacteria. They are very resistant to radiation. So Deinococcus radiodurans is very resistant to radiation. It can withstand radiations up to 1,500 times the dosage that will kill humans. That's incredible. Then you have the Thermos aquaticus. The Thermo aquaticus is a very heat-resistant bacteria. Has been isolated from environments that are over 100 degrees Celsius. And it's really applicable, very, very important for us today, because their DNA polymerase, known as Taq polymerase, is what is used in PCR because it's very heat stable. Then you have the phototropic bacteria. You have the autotrophic ones, so those that use light. So the autotrophic bacteria are grouped into 5 different groups. There are 4 phyla though, so they belong to 4 different phyla, but they are in 5 different groups. And the grouping is based on the pigments that they produce, so based on the pigments that they produce, and the source of their electrons. So that if you take blue-green bacteria, the pigments that they produce is blue-green. So they also known as cyanobacteria. What cyanobacteria uses as a source of its electrons, is water. So water is a source of its electrons. Okay. For green sulfur bacteria usually would use hydrogen sulfide as a source of its electrons. So it's the same with purple sulfur. So the pigments that are produced are the ones that are listed. The green sulfur will produce green pigment, purple sulfur will produce purple pigment as well. And then you have the green non-sulfur and the purple non-sulfur. They will use organic molecules as a source of their electrons. Organic molecules. Okay. For cyanobacteria, these are a few information that makes them distinct. They are gram-negative. They have various morphology. So as you can see in the slide, you have ones that are very spiky with a lot of thin threads. You have others that appear in pairs, more spherical. Then you have the filamental strains as well. So they reproduce by binary fission. They possess chlorophyll. They synthesize food using the photosynthetic process that is described as oxygenic. There are 2 types of photosynthesis. There is oxygenic and anoxygenic. Oxygenic photosynthesis generates oxygen at the end of the photosynthetic process. So oxygenic photosynthesis will generate oxygen at the end of the photosynthetic process. And so they are thought of as one of the organisms that help transform Earth at the very beginning. An example is of a modern kind is Prokaryotes. Prokaryotes is very common, most abundant in tropics and subtropics. and it's known to be the smallest. It's just 1 micrometer in size, the smallest photosynthesizer on Earth. Among the cyanobacteria are a few nitrogen fixers. These nitrogen fixing bacteria would be able to convert nitrogen gas into ammonium, which the cells can use. And it's only because they have an enzyme known as nitrogenase, which exists in specialized cells called heterocysts. So heterocyst cells are the ones that have been labeled H in these pictures that I have here. And what they have is that within them, they have these specialized enzymes. They have these specialized genes that synthesize the enzyme hydrogenase, which is then used to convert atmospheric nitrogen into ammonium for cells to use. Then you have the green and purple. The green and purple do not have the typical chlorophyll that exists in cyanobacteria and algae and eukaryotic plants. What they do have is known as bacterial chlorophyll. That's very related to the regular chlorophyll that we know. But they are anoxygenic, so the photosynthetic process does not produce oxygen at the end. There are sulfur and non-sulfur forms, and then based on the pigments, so you have the green sulfur, which have green pigments, and the deposits that they produce would be deposited, in terms of the pigments, would be deposited outside the cell. And then for the purple sulfur, the deposits are produced inside the cell. So these are outside the cell, as you can see in this picture on the surface. And then these are produced within the cell. Okay. So for the representative, the green reps, you have, for the sulfur groups, you have the phyla chlorobi. These are non-motile. And then for the non-sulfur, you have the chloroflexi phyla. They are filamentous in the second picture, and they're able to glide. An example would be the chloroflexox bacteria. And then for the purple, the representatives are all those that are found in the group Proteobacteria. So for our participation question for the week, I want you to state at least 2 similarities and 2 differences between green sulfur bacteria and cyanobacteria. State at least 2 similarities and 2 differences between green sulfur bacteria and cyanobacteria. And then I'm doing something different. I'm giving you an opportunity to get points for extra credit. And so there's an extra credit question. An extra credit question is that based on your knowledge of microbial genetics and mutagenic effects of radiations on DNA, what characteristics of Deinococcus radiodurans, this is it, Deinococcus radiodurans, that enables it to resist extremely high dosage of radiation? Based on your knowledge of microbial genetics and the mutagenic effect of radiations on DNA, what characteristics of Deinococcus radiodurans enables it to resist extremely high dosage of radiation? So I'm going to open a portal on Blackboard that you can post for the extra credit. So this brings us to the end of the first part of our lecture on classification of prokaryotes.